If you are walking through a dense, quiet forest right now, listening to this, I want you to just look down for a second.
Yeah, especially if it's right after a rain.
Exactly. Picture the air being really cool, maybe carrying that heavy, earthy, um, petrichor smell.
That classic damp forest smell.
Right. You're scanning the leaf litter, all those decaying pine needles and damp soil, and right there, just pushing up through the detritus, you spot a completely unassuming, pale, biscuit-colored mushroom.
It really does just look like a dropped piece of pastry.
It honestly looks like a rustic little hand pie that someone just lost on their hike.
Uh-huh.
But if you were to kneel down and pick it up, which, as we are definitely going to get into, you should be very, very careful about doing, you'd notice two incredibly strange things right off the bat.
Right, the smell and the tears.
Yeah, the smell. Instead of smelling like damp earth or that typical, mushroom funk, it smells intensely, bizarrely, like a freshly cut radish.
It has a very sharp, almost peppery scent.
And second, if you tilt it back and look really closely at its gills, it literally appears to be crying.
It genuinely looks like it's weeping. I mean, it exudes these distinct watery droplets that eventually dry up and leave these permanent brown tear stains right on the underside of the cap.
It's the strange little weeping pastry in the woods.
Yeah.
It is the poison pie mushroom, scientifically known as Hebeloma crustuliniforme.
Which is quite a mouthful.
It is. And we are spending this entire deep dive unpacking the hidden, honestly almost unbelievable world going on inside and underneath this one specific fungus. We are pulling from a massive stack of botanical research, ecological surveys, and some intensely detailed taxonomic literature that you, the listener, provided to do this.
We've got a lot of ground to cover.
We really do. We're looking at everything from its highly specific chemical weaponry to its bizarre role as an underground biological broker.
Because the broader mission here is really to challenge how we look at the mundane. On the surface, the poison pie is just a bland, medium-sized mushroom. But when you peel back the layers, when you look at its genetics, its chemistry, and the microscopic metropolis it operates, it forces you to reconsider how life in a forest actually functions.
Okay, let's unpack this. Let's start with the physical reality of bumping into one of these. Because foragers run into the poison pie all the time, right? I mean, it's a super cosmopolitan species.
Oh, very much so. If you're in a temperate region in the northern hemisphere, so across North America, Europe, parts of Asia, you are solidly in poison pie territory.
So they're everywhere.
Pretty much. You'll usually find them forming these little troops or scattered groups in deciduous woodlands, maybe under oaks or birches, but they're incredibly adaptable, so they show up under conifers as well.
Okay.
Physically, we're talking about a mushroom with a pale tan, almost buff-colored cap. It starts off convex, maybe 3 to 10 centimeters across, and as it gets older, it just flattens out like a pancake.
Right.
And the stem is cylindrical, and this is a really key identifier for any foragers listening. It completely lacks a ring.
The ring being that little skirt of tissue left over from when the mushroom was expanding out of the ground.
Exactly.
And I really have to point out the dark irony of its scientific name here. Hebeloma crustuliniforme. That specific epithet, crustuliniforme, literally derives from the Latin word crustulum, which means a small pastry or a little biscuit.
They really leaned into the bakery theme.
They did. The early mycologists looked at this thing and officially named it the mushroom shaped like a little pie, which is just a terrible public relations move considering its history. I mean, this completely innocuous food-like appearance has lured in a lot of hungry foragers over the centuries.
It looks rustic, it looks edible, it looks like something you'd want to eat.
Right.
But the visual deception is profound.
Yeah.
And it's the physical features upon closer inspection that really start to hint at its strange biology. We mentioned the tears a moment ago, but there is also a structure on the cap called the exude.
An exude. Okay, let me guess based on the prefix. Ixo usually relates to something sticky or resinous, right? Like ixidid ticks.
You're exactly on the right track, yeah. The exude is a specialized layer of hyphae, which are those microscopic fungal threads on the very surface of the cap. And in Hebeloma crustuliniforme, this layer is actually embedded in a gelatinous matrix.
A gelatinous matrix, so it's gooey.
Well, it depends on the weather. When the forest humidity drops and the air dries out, it might just feel a little bit tacky to the touch, but when the environment is wet, it becomes highly viscid. It's essentially a layer of biological slime.
So it's basically walking around in a slimy raincoat.
Functionally, yes, absolutely. The primary hypothesis in the research is that this gelatinous layer acts as a buffer against environmental fluctuation. You have to remember, a developing mushroom is mostly water, and its primary goal is to protect the billions of microscopic spores developing underneath its cap.
Right, the reproductive cargo.
Exactly. If a sudden dry spell hits, the exude prevents the underlying tissues from desiccating. It locks the moisture inside the fruiting body.
Which brings us back to the crying gills, because that seems, I don't know, totally counterintuitive to me. If it's building this advanced slime shield to keep moisture in, why is it simultaneously leaking water out of its gills?
Yeah, it sounds like a design flaw, but that process is called guttation, and it's actually one of the most debated physiological phenomena in mycology right now. When the mushroom is young and the soil is damp, the edges of the lamellae, the gills, actively push out fluid.
So it's not just dew.
No, it's important to understand this isn't just passive condensation like dew forming on a cold glass of water.
So it's an active biological process.
Yeah.
The mushroom is purposefully sweating.
Purposefully sweating is a good way to put it. The prevailing mechanical theory relates to internal cellular regulation. Plant and fungal cells operate on turgor pressure.
Turgor pressure, like water balloons.
Exactly like water balloons. You can imagine the cells like water balloons stuffed inside rigid cardboard boxes, the box being the cell wall. When the mushroom is in a rapid growth phase, it's pumping massive amounts of water and nutrients from the underground mycelial network up into the fruiting body to expand it.
Trying to inflate the balloon as fast as possible to just shove the cap out of the dirt.
Right, but if the ambient humidity in the forest is too high, water can't evaporate off the surface of the mushroom to maintain the flow. It's a process akin to transpiration in plants.
Oh, I see. The air is already too full of water.
Yes, and so the internal pressure builds up dangerously high. So, to prevent the cells from literally rupturing under the hydrostatic pressure, the fungal tissue actively secretes the excess fluid out through specialized areas on the gill edges. It's a literal pressure release valve.
Wow. Okay, that makes perfect mechanical sense. But you said earlier that the fluid dries and leaves brown spots. If it was just excess water being vented, wouldn't it just evaporate clean? What else is in that fluid?
And that is exactly where the mechanical theory gives way to ecological speculation. Because those droplets are not just water. They are complex chemical cocktails. They contain organic acids, enzymes, and secondary metabolites.
So they're brewing something in there.
They are. Some chemical ecologists argue that guttation droplets might act as a localized chemical beacon. Because remember, fungi don't have nervous systems, right? They interact with their surrounding environment entirely through chemistry.
So the tears could be laced with compounds designed to, like, attract something specific to help it, or maybe repel something dangerous?
Exactly. When you look at a weeping poison pie, you are looking at a visible manifestation of an invisible chemical factory. And we know from the sources that that factory is capable of producing some incredibly aggressive compounds, which actually brings us to the poison part of the poison pie.
Right. Let's get into the namesake, because if you were a forager and you get fooled by the biscuit-like appearance, and you completely ignore the radish smell,
Which you shouldn't.
which you definitely shouldn't, but say you do, and you chop this up and put it in a risotto, what actually happens to your body?
The regret sets in quite rapidly, honestly. The onset of symptoms usually occurs anywhere from 15 minutes to about 4 hours post-consumption.
Oh, wow, that's fast.
Very fast. And the clinical presentation is severe gastrointestinal distress. You are looking at acute nausea, violent and intractable vomiting, severe abdominal cramping, and profuse colicky diarrhea.
Oof. It is basically the absolute worst food poisoning you can imagine.
Pretty much.
But I think we need to draw a hard line here regarding fungal toxins, because the public perception of a poisonous mushroom is usually skewed by the really famous killers. You know, when people hear poison, they think of the death cap Amanita phalloides or the destroying angel.
Right, the lethal ones.
Yeah, but the chemistry of those mushrooms is fundamentally different from our little biscuit here, isn't it?
Completely different mechanism of action, and obviously a vastly different outcome. The Amanita species contain a class of toxins called amatoxins, and these are complex, ring-shaped peptide cyclopeptides.
And amatoxins are basically cellular assassins, right? They don't just upset your stomach and leave.
Far from it. Amatoxins are insidious because they pass right through the stomach and are taken up directly by the liver. Once inside a hepatocyte, a liver cell, the amatoxin physically binds to an enzyme called RNA polymerase II.
RNA polymerase II.
Yes, and this enzyme is responsible for transcribing DNA into messenger RNA.
So it literally halts the production of new proteins.
Exactly. It brings the cellular machinery to a grinding halt. The liver cells cannot repair themselves, they cannot function, and they just slowly die off. This is why amatoxin poisoning often has a delayed onset of symptoms, which is followed by a deceptive period where the patient actually feels a bit better.
The honeymoon phase.
Right before total hepatic failure and death, it's systemic structural destruction of the organ.
The Amanita quietly dismantles your organs from the inside out over a week. But the poison pie is not doing that. The poison pie is not a quiet assassin. If I'm reading these toxicological sources right, the poison pie is basically a bouncer at a club who's equipped with biological pepper spray.
The bouncer analogy works beautifully there, because the intent of the toxin isn't necessarily to kill the predator, but to forcefully and immediately eject them from the premises.
Get out right now.
Exactly. The toxins in Hebeloma crustuliniforme act as direct, highly localized irritants to the mucosal lining of the gastrointestinal tract.
It wants you out. It triggers every rapid purge reflex your body has. It's generally not lethal to a healthy adult, right? Unless they somehow lose so much fluid that severe dehydration and electrolyte imbalance become life-threatening.
Correct. But it is an incredibly aggressive, targeted defense mechanism.
So what is the pepper spray actually made of?
What's fascinating here is the active defensive agents are a unique class of secondary metabolites called hebelomic acids.
Hebelomic acids.
Yes. And specifically, toxicologists point to hebelomic acid A as the primary culprit. To understand why it hits the mammalian gut so hard, we really have to look at its structure. It's a highly oxygenated lanthanide-type triterpene ester.
Okay, let's unpack that for a second, because the lanthanide-type triterpene ester is a really dense sequence of biochemical jargon.
It is a bit of a mouthful.
Let's start with the triterpene. Terpenes are essentially the aromatic building blocks of nature, right? They're made of these repeating 5-carbon units called isoprene.
That is the foundation, yes. Plants and fungi stitch these little isoprene units together. When you stitch 6 of them together, you get 30 carbon atoms, which forms a triterpene.
Got it.
They are large, bulky, often multi-ringed lipid molecules. They actually form the structural basis for a lot of steroids and sterols in biology.
Okay, so it's this heavy, bulky, multi-ring structure. But what makes hebelomic acid A so weaponized? Why does it cause such a violent reaction?
The weaponization comes from a very specific and rare molecular attachment, what chemists call a moiety, that's hanging off that bulky triterpene core. It's called a 3-hydroxy-3-methylglutaric acid moiety, or HMGA for short.
HMGA. Wait, I actually recognize that from human metabolic pathways. Doesn't HMGA have something to do with how we make cholesterol?
You have a good memory. You are recognizing the mevalonate pathway. In mammals, including humans, the mevalonate pathway is the fundamental metabolic engine that produces cholesterol. And a really critical intermediate step in that biological assembly line involves HMGA.
Wait, so you are telling me this unassuming little woodland mushroom is somehow synthesizing a byproduct of the mammalian cholesterol pathway and just bolting it onto a heavy terpene molecule to use as a weapon?
It is a profound example of biochemical hijacking. The fungus has evolved the enzymatic machinery to synthesize this HMGA moiety and attach it via an ester bond to the triterpene. That highly specific, highly oxygenated structure is the key.
Because it fits perfectly into the receptors in our gut.
Exactly. It interacts violently with the mammalian system. And this specific molecular architecture is also what gives the poison pie its intensely bitter taste. It's what creates that raw, penetrating radish odor we talked about.
Oh, the radish smell comes from that.
Yes, those are volatile organic compounds off-gassing from the breakdown of these chemical precursors. It's an evolutionary masterclass in deterrence. The mushroom advertises its toxicity with a bitter taste and an odd smell, and then it backs it up with a cholesterol-derived triterpene that causes immediate, violent regret.
So we have a mushroom that disguises itself as a rustic little biscuit, covers itself in a humidity-regulating slime coat, actively regulates its internal pressure by weeping chemically complex tears, and defends itself with a highly oxygenated cholesterol pathway-derived molecular weapon.
It's quite the resume.
It really is. You would think that an organism with a chemical signature this specific, this highly evolved, would be incredibly easy for biologists to classify. Like you find the radish-smelling weeping biscuit, you test it, you categorize it, done.
You would think so. But the assumption that morphological and chemical uniqueness translates to an easy taxonomy is a trap that mycology actually fell into for over a century.
And it led to a massive scientific illusion. I really want to shift into the history here. because this is where the story of the poison pie basically becomes a detective novel.
Hmm.
Historically, what was happening when early naturalists tried to map this mushroom?
The history goes way back to the dawn of formal taxonomy. In 1787, a French mycologist named Jean-Baptiste François Pierre Bouillard first described and illustrated what we now call the poison pie.
1787.
Yes, but back then, mycology was a profoundly chaotic science.
I mean, they didn't have microscopes, let alone DNA sequencers. They were basically just wandering around the woods of Europe, writing down what things look like, smelling them, and occasionally and dangerously taking small bites to see how they tasted.
Which is terrifying. But yes, the diagnostic criteria were entirely macroscopic. Bulliard and the generations of taxonomists who followed him, like Elias Fries, relied on visual syndromes. And because the poison pie is a very successful, widely distributed mushroom, it exhibits a lot of subtle, natural variation.
Right, like local adaptations.
Exactly. Sometimes it's a bit more tan, sometimes the stem is a bit thicker, sometimes it whoops more profusely depending on the local weather.
So how did they handle those slight differences without genetics?
They did what human brains naturally do. They generalized. The name Hebeloma crustuliniforme basically became a taxonomic junk drawer.
A junk drawer.
Yeah, for the next 150 years, if a field mycologist found a pale, medium to large agaric mushroom that lacked a ring on its stem, smelled distinctly of radish, and had brown spots on the gills, they just didn't look much further. They tossed it straight into the Hebeloma crustuliniforme bin.
It was the ultimate default setting. Looks like a biscuit, smells like a radish, makes you throw up. Put it in the poison pie folder. Next.
Exactly. It was treated as a single, widely distributed, morphologically variable megaspecies. It was a monolithic concept that dominated the textbooks right up until the late 20th century.
But then we learned how to read the source code. Molecular phylogenetics entered the chat. And we didn't just have to rely on visual approximations anymore. We could look at the actual genetic blueprints.
And that changed everything. The advent of DNA sequencing, specifically looking at the internal transcribed spacer, or ITS, regions of fungal ribosomal DNA, completely shattered the monolithic view of the poison pie.
The ITS region.
Yes, the ITS region is fantastic for fungi because it acts like a molecular barcode. It mutates at a very steady rate, allowing scientists to see exactly how closely related different specimens actually are.
So they finally scanned the barcode of the poison pie.
They did. When modern geneticists began gathering herbarium specimens from all over the world that had been confidently labeled as Hebeloma crustuliniforme for decades, and they ran that DNA through a sequencer, the results were staggering.
The sources outline this, and it completely broke my brain. They found that the poison pie is not one species. In northwestern Europe alone, what scientists thought was a single mushroom for 2 centuries is actually a complex of at least 22 distinct biological species.
22 genetically isolated entities existing in just one geographic region, all hiding under the exact same name.
I really have to push back on the mechanics of this because it challenges my basic understanding of evolution. If I take 22 distinct species of mammals, say a tiger, a house cat, a lion, a leopard, and so on, they are all felines. They share an evolutionary ancestor, but they look drastically different.
Right.
They have adapted different sizes, different coat patterns, different skull shapes. How in the world can 22 completely different biological species of this mushroom look exactly the same to experts for 100 years?
It's a great question. And to resolve that paradox, we have to introduce the concept of cryptic species and explain how a mycologist actually defined what a biological species is using intercompatibility groups.
Okay, let's walk through it. What makes something a cryptic species?
A cryptic species complex is a group of individual species that are entirely distinct and isolated from a genetic and reproductive standpoint, but they exhibit no morphological differentiation.
So they look identical.
To the human eye, and even under a standard light microscope, they are completely indistinguishable. The differences are written entirely in their DNA and their biochemistry.
So they are genetically divergent, but physically identical. But how do we prove they are actually different species if we can't tell them apart visually? Is the DNA barcode enough to just declare them separate?
The DNA barcode gives us the map, but the biological proof comes from actual reproductive testing. In these types of fungi, the sexual Basidiomycetes, the gold standard for defining a species is the biological species concept, Can two individuals mate and produce viable, fertile offspring?
So scientists were playing matchmaker in a petri dish.
Quite literally, yes. Researchers perform extensive sexual intercompatibility tests. They take spores from all these different poison pie collections and germinate them to create monospore cultures.
Monosphere cultures.
Yeah, these are haploid mycelium networks, meaning they only have half a set of chromosomes, much like a human sperm or egg cell.
Ah, so they're looking for a mate to complete the set.
Exactly. Then the researchers physically place two different haploid cultures next to each other on an agar plate and just watch what happens. If they belong to the same species, the microscopic hyphal threads will reach out, physically fuse together in a process called plasmogamy, and exchange nuclei to form a stable, reproducing, diploid organism.
But when they did this with the 22 different versions of the poison pie.
They refuse to fuse.
Wow.
The researchers discovered 22 distinct intercompatibility groups, or ICGs. The cultures from group 1 would only fuse with other cultures from group 1. If you placed a group 1 next to a group 14, they might touch, but the cellular recognition pathways would just reject the pairing outright.
They know they aren't the same.
They do. They are reproductively isolated. They are true, distinct evolutionary lines.
That perfectly answers the how we know part.
Yeah.
But it doesn't really answer the why. Why haven't they changed their outfits? If they have been genetically separated long enough that their cells literally reject each other, why do they all still look like the exact same tan biscuit with weeping gills?
Because evolution is blind, and it only acts on what is strictly necessary for survival. The reason they look the same is that speciation within the Hebeloma crustuliniforme complex is an ongoing, highly active, and relatively recent evolutionary explosion.
Recent in evolutionary time, meaning maybe hundreds of thousands or a few million years rather than tens of millions.
Exactly. Genetic divergence, the mutation of their DNA and the shifting of their cellular mating recognition proteins, is happening at an incredibly rapid pace. This reproductive isolation is outstripping their morphological differentiation.
So they just literally haven't had the time to evolve different cap shapes or colors.
More importantly, there is no evolutionary pressure pushing them to change how they look. Think about it. A tan, convex cap with the slimy exude clearly works perfectly well for survival in a temperate forest.
Right.
Evolution doesn't invent new shapes just for the sake of variety. Taxonomists refer to this as a stable morphological syndrome.
If the cap ain't broke, don't fix it.
Precisely, but that leads to the ultimate driver of this cryptic diversity. If the evolutionary pressure isn't on their physical shape, what is driving this frantic genetic mutation? Why are they splintering into dozens of isolated species in the first place?
Well, whenever there's an evolutionary arms race, it's usually about food or real estate. And reading ahead in the sources, the answer lies beneath the soil. It's about who they are partnering with. They are rapidly adapting to interface with entirely different species of trees.
And that shift from taxonomy to ecology reveals the true nature of the poison pie. These mushrooms are the architects of the forest's underground economy. They are obligate ectomycorrhizal fungi, or ECM fungi.
Let's draw a clear boundary here for the listener, because there are broadly different ways that fungi make a living in the world. We are generally used to thinking of fungi as the cleanup crew, the saprotrophs.
Right. Saprotrophic fungi are the decomposers. They are the organisms that land on a fallen oak log, a pile of dead autumn leaves, or an old piece of fruit. They secrete powerful hydrolytic enzymes out of their mycelium to chemically dismantle the dead organic matter.
Just melting it down.
Exactly. They break down tough plant polymers like cellulose and lignin, absorb the freed carbon, and return the rest to the soil. They are nature's recyclers.
They eat the dead. But the poison pie and its 22 cryptic siblings do not do that. They don't care at all about the dead log.
No. ECM fungi, like Hebeloma crustuliniforme, engage in a mutualistic living partnership with the roots of actively growing, forest-forming trees. We are talking about the major canopy builders, the oaks, the pines, the birches, the trembling aspens.
The giants.
Yes. The mushroom doesn't just live in the general vicinity of these trees. It physically and intimately fuses its body with the tree's living root system.
I always try to visualize what this looks like at a microscopic scale, because the concept of a fungus merging with a giant oak tree sounds like science fiction to me. It's an interkingdom physical connection. How does the mushroom actually dock with the tree?
The docking station is a structural marvel. It's known as the Hardig net. It begins when the fungal hyphae, those microscopic exploring thread-like tubes, detect the chemical signals of a compatible host tree's lateral roots.
The lateral roots, so the fine, hair-like feeder roots of the tree, not the massive wooden anchor roots we trip over.
Right, the very fine tips. The fungus approaches these fine roots and begins to weave an incredibly dense, multi-layered sheath of fungal tissue completely around the outside of the root. This is called the mantle.
Okay.
It essentially acts as a protective glove over the root tip, but it doesn't stop there.
It goes inside.
It does. The hyphae begin to push inward. They penetrate the outer layers of the plant root, slipping between the epidermal and the cortical plant cells.
Wait, just to clarify,
Yeah.
they don't stab into the plant cell itself like a parasite. They don't rupture the cell wall.
No, and that distinction is absolutely crucial. If they ruptured the cell wall, the plant's immune system would instantly trigger a massive defensive response. Instead, they navigate the interstitial spaces, weaving tightly between the plant cells to form a highly complex, interlacing microscopic labyrinth.
Like weaving a basket between the cells.
Exactly. This labyrinth of fungal threads weaving through plant tissue is the Hardig net.
It's basically maximizing surface area.
Right.
By weaving between all the individual plant cells, you create this massive microscopic interface where the fungus and the tree are practically touching over a huge area.
And that immense surface area is the stage for the most consequential biological transaction in terrestrial ecology. If we connect this to the bigger picture, it is the engine of the Wood Wide Web.
The great trade. Let's break down the economics of this. What is the fungus bringing to the table, and what is the tree bringing?
Well, the foundational problem for a tree is that while it has access to essentially unlimited solar energy above ground, the soil below is a highly competitive, nutritionally scarce environment.
It's crowded down there.
Very. Nutrients like nitrogen and phosphorus are often locked up in complex organic molecules, or they are distributed in microscopic pockets far away from the tree's roots.
And tree roots, even the fine ones, are relatively thick and clunky on a microscopic scale. They can only reach so far. The sources talked about the depletion zone. Basically, a tree root acts like a straw. It sucks up all the available water and dissolved nutrients immediately surrounding it, creating a barren zone. To get more food, the tree has to physically grow the root longer, which takes a lot of energy and time.
That is exactly where the fungus provides an invaluable service. Fungal hyphae are magnitudes thinner than plant roots. A single teaspoon of forest soil can contain miles of these microscopic fungal threads.
Miles.
In a teaspoon.
Yes. The fungus acts as a sprawling, highly efficient, hypersensitive scavenger network. It explores far beyond the plant's depletion zone, squeezing into microscopic pores in the soil that the tree could never dream of reaching. I picture the fungus as an outsourced, highly specialized mining corporation. The tree is this massive solar-powered factory above ground. It's manufacturing pure, liquid, carbon glucose and sugars using sunlight and carbon dioxide. But to build proteins, to build DNA, the tree factory needs raw mineral resources, specifically nitrogen, from deep underground.
Right.
And its own roots are too inefficient, so the oak tree hires the Poison Pie Mining Corporation.
It's a very fitting analogy. The mushroom network goes out into the dark, mines the nitrogen, transports it miles back through its hyphal tubes, and brings it to the loading dock, the Hardig net. And the tree pays for the shipment in pure photosynthetic sugar.
The exchange is incredibly highly regulated. And to understand just how vital and how specialized this fungal mining operation is, we have to look at a specific experiment detailed in the sources involving the trembling aspen, Populus tremuloides.
Yes, the nitrogen flavor experiment. That study is fascinating.
Walk us through how researchers isolated what the poison pie was actually doing for these trees.
The researchers wanted to see if the fungus was just acting as a generic fertilizer hose, or if it was fundamentally changing how the tree interacted with soil chemistry. So they took trembling aspen seedlings and grew them in highly controlled environments. Half the seedlings were inoculated with the Hebeloma crustuliniforme fungus, so they were allowed to form that Hardig net partnership.
Okay.
The other half were kept sterile as a control group, growing entirely on their own.
Okay, so partner trees versus solo trees.
Then the researchers manipulated the primary resource, nitrogen. But nitrogen in soil doesn't just come in one form, it exists in different molecular structures. The two main inorganic forms available to plants are nitrate and ammonium.
Nitrate being a nitrogen atom bound to oxygen, and ammonium being a nitrogen atom bound to hydrogen. They're different molecular keys.
Precisely, and plant biology has distinct preferences. The trembling aspen on its own has evolved enzymatic machinery that is heavily biased toward processing nitrate. If you give a solo aspen seedling a soil rich in nitrate, it does relatively well. But if the soil is primarily ammonium, the solo tree struggles to process it efficiently.
It's like having a car that runs great on premium unleaded, but sputters and stalls out if you try to put diesel in it.
Great analogy. What the researchers discovered was that the poison pie fungus possesses the exact opposite proficiency. The fungal mycelium is exceptionally good at enzymatically recognizing, absorbing, and assimilating ammonium from the soil.
So what happened when they put the two together in the dirt?
The results were dramatic. The trembling aspen seedlings that were partnered with the poison pie grew significantly larger with greater biomass, regardless of whether the researchers fed them nitrate or ammonium.
That is amazing. The fungus effectively bolted an ammonium processing engine onto the tree's existing nitrate processing chassis.
By forming this symbiosis, the tree fungus superorganism was no longer limited by the specific chemical state of the nitrogen in the soil. The plant could pull the nitrate, the fungus could mine the ammonium, and together they exploited the soil's resources with a level of efficiency that neither could achieve independently. It's a perfect illustration of why the forest canopy is entirely reliant on the fungal networks beneath it.
It is brilliant ecological synergy. But whenever we talk about evolution, there is always a catch. Nature doesn't hand out superpowers for free. It sounds like the mushroom is doing all the heavy lifting down in the dark. It is building the network. It is synthesizing the enzymes. It is mining the ammonium. But what did the mushroom have to give up to get this good at its job?
And here's where it gets really interesting. The cost of this symbiosis is one of the most fascinating aspects of fungal evolution. To become an indispensable partner to the tree, Hebeloma crustuliniforme literally had to burn its boats.
I love this concept. Explain what burning the boats means genetically.
Earlier, we discussed the saprotrophic fungi, the decomposers that eat dead wood by secreting plant cell wall-degrading enzymes, or PCWDEs.
Right.
Evolutionary biologists believe that the ancestors of ectomycorrhizal fungi like the poison pie were originally saprotrophs. They were independent, they lived in the soil, broke down dead leaves, and gathered their own carbon.
They were self-employed.
They were. But as these ancestral fungi began to experiment with symbiosis, perhaps initially just scavenging leaked sugars around plant roots and eventually forming the full Hardic net, they found themselves tapping into a massive, highly reliable pipeline of pure plant glucose.
A pipeline powered by the sun. It's essentially unlimited energy as long as the tree is alive.
Exactly. But maintaining a massive genome is energetically expensive. If a fungus is constantly receiving easy sugar from a tree, the genes responsible for producing the complex, heavy enzymes needed to digest dead wood become redundant.
Use it or lose it.
Literally. Over millions of years of evolution, a phenomenon called genetic reduction occurred. The ancestors of the poison pie simply lost the genetic code required to produce PCWDEs. The genes degraded, mutated, and were ultimately scrubbed from the genome entirely.
So the mushroom physically deleted its ability to feed itself. It arrived on the shores of Symbiosis, looked at the steady supply of tree sugar, and burned the boats of independence.
It is an absolute obligate dependency now. If you take a mature poison pie mycelial network and disconnect it from a living tree root, it cannot simply revert back to eating dead leaves. It lacks the genetic hardware. It will literally starve and die in a forest full of organic carbon.
That is an incredible evolutionary gamble, but clearly it paid off because, as we established, the poison pie is everywhere. But if it isn't spending its metabolic energy building enzymes to break down carbon anymore, what is it doing with all that extra processing power?
It reallocated that genetic real estate to become a master of nitrogen acquisition. Since it can't digest complex carbon, its enzymatic arsenal is almost entirely dedicated to degrading soil organic nitrogen. It produces highly specialized extracellular proteases.
Okay, let's break that down. Extracellular, meaning it secretes them outside of its body into the dirt. And a protease is an enzyme that acts like molecular scissors, right? It cuts up proteins.
That's a perfect visualization. The soil is full of organic nitrogen trapped in large, inaccessible protein molecules, things like dead insect exoskeletons, decaying microbial matter, complex organic polymers.
Stuff the tree can't touch.
Exactly. The tree roots cannot access this. But the fungal hyphae secrete these proteases into the soil. The enzymes snip the complex proteins into smaller, soluble amino acids and peptides, which the fungus then absorbs.
But wait a minute. If the fungus is entirely reliant on the tree for its carbon energy, how does it control when it fires up this mining operation? It takes energy to build and secrete those protein scissors. If it just pumps them out constantly, it might waste all the sugar the tree just gave it.
The regulation of these enzymes is a marvel of biochemical engineering, driven by a highly calibrated two-part feedback loop. The fungus is constantly sampling its environment. First, it monitors the soil. If the hyphae detect that there is already an abundance of simple, easily absorbable inorganic nitrogen, like the ammonium we discussed earlier, the genetic transcription of the proteases is chemically repressed.
So the fungus says the gold is already lying on the ground. Don't waste energy building excavators. It shuts off the enzymes.
Precisely. It is extremely conservative with its energy. But the second part of the feedback loop is where the symbiosis really shines. The production of these nitrogen-mining proteases is actively stimulated by the presence of glucose flowing from the host tree.
Oh, wow. The tree sugar isn't just food. It's a chemical signal that turns the factory on.
Yes. The sugar initiates a signaling cascade within the fungal cells that upregulates the protease genes. This ensures a perfect supply and demand equilibrium.
That is a biological union contract enforced by genetics. Think about it. The fungus will only expend the massive metabolic effort required to mine organic nitrogen when the tree is actively paying it in carbon. If the tree gets sick or the canopy gets shaded out and the tree stops sending sugar down the roots.
The fungus senses the drop in glucose, the stimulation ceases, the protease production drops, and the nitrogen mining operation shuts down. The transaction must remain equitable. Neither partner can be parasitized by the other without collapsing the entire leaf.
It is stunning. I mean, we've taken this little pale crying biscuit mushroom and we've uncovered an organism that synthesizes modified cholesterol to cause violent human illness, hides 22 divergent species under an evolutionary cloak, and operates a genetically regulated, obligate mining network that sustains the giants of the forest.
And we haven't even reached the deepest layer of its ecology, because the poison pie isn't just a partner to the tree canopy above it. The fruiting body itself, the mushroom sitting on the forest floor, is actually acting as a landlord to an entirely separate microscopic metropolis.
This is where we need to return to the velvet rope at the Poison Pie Club. Let's go back to section 1 where we talked about those intense, bitter toxins, the hebelomic acids. We established that if a deer, a squirrel, or a human comes along and tries to eat this mushroom, the highly oxygenated triterpenes act like chemical pepper spray, violently rejecting the mammal's digestive system. But in reading the entomological sources you provided, I noticed an incredible loophole in this defense system.
The defense is targeted, but it is not universal. While the toxins are highly effective against generalist mammalian foragers, there is a specific lineage of insects that has completely bypassed the security system. We are talking about fungus gnats, primarily from the family Mycidophilidae.
The sources specifically call out a species named Mycetophila ichneumoniae. What is their relationship with the poison pie?
Mycetophila gnats are obligate mycetophages, meaning their entire life cycle revolves around eating fungi. But M. ichneumoniae is highly specialized. The adult females actively seek out the fruiting bodies of Hebeloma species to lay their eggs.
Okay.
When the larvae hatch, they begin to voraciously consume the fungal tissue.
But the tissue is pumped full of hebelomic acid A. Why aren't the insect larvae violently ill? How do they survive eating the pepper spray?
They have evolved a physiological immunity. While the exact detoxification pathways are still being studied, these specialized larvae likely possess specific cytochrome P450 enzymes in their gut that can rapidly bind to and neutralize the triterpene toxins before they cause any cellular damage.
They literally evolved an antidote.
More than that, they have weaponized the mushroom's own defense against the rest of the insect world. The gnat doesn't just tolerate the poison, it relies on it.
Because the poison keeps everyone else away. It's the velvet rope. Think about a regular non-toxic mushroom in the forest. It's a free buffet. You have slugs, beetles, millipedes, and dozens of fly species all competing to lay eggs and eat the tissue. It's a war zone. But the poison pie's hebelomic acid kills or repels all those generalist competitors.
The mushroom provides a protected, heavily defended nursery. By evolving the specific enzymes to neutralize the toxin, the Mycetophila gnats gain exclusive access to a massive carbon resource. They have a completely safe, competition-free haven to rear the next generation.
It's an evolutionary hack. But as wild as the insect nursery is, the guest list goes much, much deeper. Because the most mind-bending revelation in the sources was about what is happening on a bacterial scale inside this mushroom.
This raises an important question, and it moves us into the realm of the fungal microbiome. For a long time, mycologists largely treated the inside of a healthy, growing, mushroom-fruiting body as a relatively sterile environment. But recent genomic sequencing has revealed that the sporocarp, the physical body of the poison pie, is teeming with life.
It's crowded.
It is. It hosts a highly complex, structured community of what researchers call fungus-associated bacteria, or FAB.
How many bacteria are we actually talking about here?
Well, in single specimens of Hebeloma crustuliniforme, genomic assays have isolated and identified at least 56 distinct active strains of bacteria living entirely within the fungal tissue.
56 different bacterial strains living inside a single mushroom cap. What are they doing in there?
Yeah.
Because bacteria are opportunistic. If they're living inside a mushroom, they're either infecting it as pathogens or they're getting something out of it.
It is a profoundly synergistic relationship. When geneticists analyzed the genomes of these specific 56 bacterial strains, they found that the bacteria were heavily enriched with two very specific types of gene clusters. First, genes for motility.
Meaning they have little flagella, they can swim.
Yes, they aren't just sitting passively trapped in the tissue, they are actively navigating through the interstitial fluids between the fungal hyphae within the mushroom.
Okay, swimming bacteria. What's the second gene cluster?
Complex carbohydrate metabolism. But not just any carbohydrates. When a plant builds structure, it uses cellulose. Fungi don't use cellulose. They build their cellular walls out of chitin, the exact same rigid polymer found in the exoskeletons of crabs and beetles.
Right.
Furthermore, fungi store their energy using a very specific, unique disaccharide sugar called trehalose.
So let me guess. The bacteria inside the poison pie have the exact enzymatic tools required to digest chitin and trehalose.
Precisely. They have evolved specialized metabolic pathways to feed exclusively on fungal carbon sources. The mushroom is literally cultivating a bacterial colony within its own body, providing them with a secure habitat again, protected from outside predators by the mushroom's toxicity, and feeding them a specialized diet.
But nature is an economy. Nobody rides for free, especially not in a highly regulated symbiotic system like this. If the mushroom is spending its hard-earned tree sugar to synthesize trehalose just to feed 56 strains of bacteria, what is the mushroom extracting from the bacteria in return?
The answer connects the entire micrometropolis back to the massive forest canopy. Genomic profiling reveals that a significant portion of these fungus-associated bacteria possess the NIF-H gene complex.
The nitrogenase genes. They're nitrogen fixers.
They are capable of biological nitrogen fixation. They can take inert dinitrogen gas from the microscopic air pockets inside the mushroom and the surrounding soil, break the incredibly strong triple bond between the nitrogen atoms, and convert it into bioavailable ammonia.
I need to synthesize this because the scale of this loop is just staggering. We started this deep dive talking about the great trade in the wood wide web. The entire reason the tree partners with the poison pie, the reason the fungus deleted its ability to eat dead wood, is so the fungus can act as a massive nitrogen mining corporation for the tree.
Yes, that is its obligate ecological role.
But nitrogen is incredibly hard to acquire. Even pulling it out of the air takes massive amounts of energy. So the mushroom doesn't just rely on soil scavenging, it literally imports a workforce. The mushroom takes the sugar it gets from the oak tree. It turns that sugar into fungal trehalose. It feeds that trehalose to a swimming colony of 56 bacterial strains living inside its toxic cap.
Yeah.
The bacteria use that energy to chemically fix nitrogen out of the air. The bacteria excrete the nitrogen, the mushroom absorbs it, and then shifts it miles underground back to the oak tree to pay for more sugar.
It is a nested Russian doll of symbiosis. The bacteria feed the fungus feeds the tree feeds the fungus feeds the bacteria, and the entire operation is guarded by insects immune to a localized biological weapon derived from cholesterol.
It is almost too complex to visualize. You are walking in the woods, you see a boring, pale, unassuming mushroom sitting in the dirt. You might lightly kick it with your boot or just walk completely past it without a second thought. But what you just walked past is not a vegetable.
Far from it.
It is a highly armed chemical fortress. It is a secure, velvet rope nursery for specialized gnats. It is a bustling, microscopic city of nitrogen-fixing bacteria. And it is a vital physical extension of the root system of the massive 100-foot tree towering above your head, constantly negotiating chemical supply chains across the forest floor.
It completely dismantles our macrocentric view of nature. It proves that the real architecture of an ecosystem isn't what we see above ground. It is the invisible, microscopic, chemical negotiations happening beneath our feet and within the tissues of organisms we dismiss as mundane.
So what does this all mean? We've gone from a toxic biscuit to a master of evolutionary disguise hiding 22 cryptic species to a vital ecological organ. What does this all mean for how we understand biology?
It leaves us with a deeply philosophical challenge regarding the nature of identity. The concept of the holobiont suggests that we shouldn't look at an organism as a single isolated entity, but as a vast assemblage of a host and its microbiome.
Like how a human isn't just human DNA, but also the billions of gut bacteria that help us digest food and regulate our moods.
Exactly, but taken to an extreme. When you look down at a poison pine mushroom on the forest floor, I want you to ask yourself, what are you actually looking at? Where does the individual begin and end?
It's blurry.
Very. If this fungal organism has genetically deleted its ability to feed itself and cannot survive a day without the tree's photosynthetic sugar, if its physical root system is so intimately interwoven and fused with the plant's cellular structure in the hardy gnat that they share cytoplasm, and if its very physical body is fundamentally dependent on an internal colony of 56 different bacterial strains to process the vital nitrogen it trades for its life.
Is it even a distinct organism anymore?
Is it an individual? Or is the biological reality far blurrier? Is it more scientifically accurate to view the poison pine not as a standalone creature, but simply as a localized temporary fruiting appendage of the forest itself? A biological node in a much larger, impossibly complex, interconnected superorganism.
That is an absolutely wild thought. Is the mushroom just an organ of the forest? It makes you realize how little we actually understand when we look at the natural world. It is something to chew on. Metaphorically, of course, because as we established with the Hebelomic acids, do not actually chew on the poison pie. Thank you to the listener for providing these incredibly dense, revealing sources and sending us down this subterranean rabbit hole. The next time you walk through the woods, keep looking beneath the surface, because clearly, the real story is always hiding entirely out of sight.